Good morning everyone. Today our topic is horse and vector, which present by me, Khor, Hoong, CHING, and Pei. What is horse? Horse is the larger participant in symbiotic relationship, serving as home and food source for smaller symbionts, which is vector. The horse is available for protein expression. Example in horse, in prokaryote is E. coli. In eukaryotes are yeast, plant, insects, mammalian cells. What is vector? Vector is an organism that transmits pathogen from one host to another. In recombinant technology, vector is a DNA molecule that used as a carrier to transport foreign DNA to another cell. It can be replicated and expressed. When vector plus foreign DNA, it is called as recombinant DNA. There is 4 types of vector: plasmid, bacteriophage, cosmid, and artificial chromosome. Plasmid is known as extrachromosomal DNA. It is a small circular double-stranded DNA molecule that carries accessory gene separated from a bacterial chromosome. Plasmid used to clone DNA ranging in size in 1 bp to 20 kbp. It is the most commonly used as bacterial cloning vector. It has 3 characteristics: origin of replication, selectable marker, and restriction site. Origin of replication can allow the plasmid replicate itself other than bacterial chromosome. Selectable marker, for example, antibiotic resistant gene, allow the plasmid to be isolated and selecting easily from the screening process. Restriction site allow the restriction enzyme to cut at the place to insert the foreign DNA. The example of the first plasmid used as cloning vector is PBR322. This is PBR322. From the figure, you can see that the origin of replication is in the center. It can replicate away from the bacterial chromosome, and the blue color thing is the restriction site, the restriction enzyme cut. There are 5 types of plasmid: fertilitative plasmid, resistant plasmid, co-plasmid, degradative plasmid, and virulent plasmid. When plasmid and gene of interest cut by restriction enzyme, the gene of interest will insert into the plasmid and ligation happen. Then this recombinant plasmid transform into the bacterial chromosome, become recombinant bacteria. The advantage of using plasmid as vector: double-stranded DNA can replicate apart from the chromosome of host cell. It is small size, which is easily take up smaller pieces of DNA. It has high copy number, which plasmid replication can be relaxed and stringent. The selectable marker allow hosts that contain it can be isolated, for example using blue-white screening, which we'll explain later. The large number of plasmid of restriction site allow different kind of restriction enzyme to cut. Blue-white screening. Plasmid that contain LAC-Z gene are able to produce beta-galactosidase, which show the blue colony on the F skull of agar plate. If the agar plate has blue colony, means this plasmid can produce beta-galactosidase. It has functional LAC-Z gene and no foreign DNA is inserted into the plasmid. If the F skull produce white colony, means no beta-galactosidase and inactive LAC-Z gene. This bacterial plasmid had take out foreign DNA into itself, and then this is a successful recombinant. This advantage of using plasmid as a vector is because plasmid is very small, so have limited capacity to accept large DNA fragment. Transformation of bacterial cell with plasmid is inefficient as the plasmid site increases. Means the larger the plasmid site, the more hard for bacteria to transform. Now I will move to bacteriophage.
The second vector that I would like to talk about is bacteriophage. Bacteriophage was discovered by bacteriologist Frederick Todd and microbiologist Felix D. Harriel. Bacteriophage, it is also known as phage or bacterial virus and classified as virus family. Bacteriophage is a group of viruses that infect bacteria, meaning it is a bacteria eater. When the phage hits a suitable bacterium, they multiply efficiently. There are several types of bacteriophage. The first one that I would like to explain is the lambda bacteriophage. Lambda bacteriophage has a linear genome of 48.5 kbp. Each end of this lambda bacteriophage consists of cohesive sites, which is approximately 12 bp. The central region are not essential, therefore it can be replaced by foreign DNA that have a size up to 23 kilobase pair. This is the overall picture of lambda bacteriophage. Both cohesive sticky ends will be joined circularization. Another type of bacteriophage is M13. M13 fish consists of circular single-stranded DNA with the size of 6.7 kilobase pair. This fish replicates in E. coli as double-stranded circular DNA. Recombinant M13 fish can either produce double-stranded replication form which can be isolated and used as plasmid, or single-stranded DNA can be isolated from fish growth medium. Example of M13 fish is M13, ampicillin 18 and 19. Now, let me explain how M13 fish undergoes cloning. First, the M13 fish, for example M13 ampicillin 18, is transfected into E. coli cell. Then plating is done and plaques are usually formed. The plaques usually consist of slow growth rather than cell lysis and of infected cells. Finally, blue-white screening using multiple cloning sites or like ZAC is done. This picture shows how the DNA is inserted into the fish. Firstly, the restriction enzyme will cut the lambda fish at the restriction site. At the same time, splicing of foreign DNA also occurs. The foreign DNA is now joined with the lambda fish DNA. Finally, in vitro packaging of recombinant molecule happens and inserted into the lambda fish which act as hosts. There are two types of cell cycle that bacterial phage can undergo. There are lysogenic and lysolytic cycle. The beginning of the steps are the same for both cycles. The first step is attachment of bacterial phage on the wall of host cell. Then the recombinant DNA is circularized inside the host cell. Here comes the splitting of the pathway. The lysogenic, the bacterial phage DNA is integrated into host DNA. Then cell division occurs and the lambda DNA replicates together with the host chromosome. However, there are some differences in lysolytic cycle. Lysolytic cycle requires the synthesis of viral proteins that are needed to form a new virus. Next, replication occurs and the lambda fish is packaged into complete virus. Finally, the cell lyses and occur to release the large number of new virus outside the host. These are the differences between lysogenic and lysolytic cycle. The advantage of bacteriophage as vector are bacteriophage is efficient in packaging and delivery mechanism. It can be carried in large insert DNA up to 24 kbp and is a very stable vector. However, there are disadvantages of using bacteriophage as vector too. The large size of DNA, which is up to 24 kilobase pair, will complicate the restriction analysis of foreign DNA. It is also difficult to produce large quantity of fish DNA compared to plasmid, and the insertion of foreign DNA is difficult to be confirmed.
Next, I will talk about cosmic. What is cosmic? Cosmic is a plasmid vector introduced by Collins and Horn that contains a lambda phage COS sequence. The COS site is the only requirement for DNA to be packaged into phage particle. Cosmic contains bacteria origin of replication, COS sites, antibiotic gene, recombinant site, and restriction endonuclease recognition sequence for cloning. Next is the cloning in cosmic vector. Firstly, COS DNA is cloned into plasmid to form cosmic vector. Next, cosmic and DNA fragments are ligated. They will undergo in vitro packaging to form phage coats. And lastly, recombinant cosmic replicates as plasmid in host cell. Why cosmic is so important? Cosmic is important because cosmic can contain large amount of DNA. It replicates as plasmids if they have a suitable origin of replication. It also contains a gene for selection such as antibiotic resistance so that the transformed cells can be identified. It can be packaged in phage caspids, which allow the foreign genes to be transferred into or between cells by transduction. Why COS sites is important? Plasmid will become unstable after a certain amount of DNA has been inserted into them, as their increased size is more conducive to recombination. To circumvent this, phage transduction is used instead. However, only COS site is able to undergo phage transduction. In this way, they are similar to using the lambda phage as vector, but only that all the lambda genes have been deleted with the expansion of the COS sequence. The next one is COS sequence. What is a COS sequences? COS sequences consist of 200 base pairs long and essential for packaging. COS sequence contain a COS N site, COS B site, and COS Q site. COS N site is a site where DNA is nicked at each strand. It has 12 base pair apart by terminates. This causes linearization of the circular cosmic with two cohesive or sticky ends of 12 base pair. The DNA must be linear to fit into a fish head. Next, COS B site, where it holds the terminates while it is nicking and separating the strands. And lastly, COS Q site is which it is held by the terminates after the previous cosmic has been packaged to prevent degradation by cellular DNAs. The advantages of using cosmic as vector is efficient packaging system of lambda for large-size DNA to prepare large genomic libraries. Next, since phage particles can accept between 38 and 53 kb of DNA, and since most cosmic are about 5 kb, so between 33 and 48 kb of DNA can be cloned in this vector. Cosmic vector can carry up to 45 kb, whereas plasmic and lambda phage vector are limited to 25 kb only. The disadvantages of using cosmic as vector is difficult to prepare large DNA fragments suitable for ligating into high molecular weight form. There is also instability of clone eukaryotic sequences due to recombination and deletion of portion of insert DNA in prokaryotic whole cell.
Okay, now let us move to the fourth vector, which is artificial chromosome.
Okay, I'm going to talk about artificial chromosome, which is short for AC. Artificial chromosome is a synthetic chromosome we use to conceal fragments of foreign DNA to integrate into the host cells. The artificial chromosome is normally used as gene mapping and modeling genetic disease. Here's how the artificial chromosome is made. First, the DNA sequence is written into the computer. Then the DNA synthetic machine process and create a short corresponding sequence. Next, the PCR is used to stitch sequence into the strand and cell with several strands into the longer base pair chunk. At last, the chunks are recombined into the original chromosome until it is completely synthetic. Next, there is two common types of artificial chromosome. The first one is bacterial artificial chromosome, which is based on the fertility plasmids of bacteria. Next one is yeast artificial chromosome, which is based on the DNA of a yeast. This table shows the different type of AC with different host cells and insert capacity. The PAC and BAC had a common host cell which is E. coli, and they can carry up to 300 kilobase pair. Next, the yeast artificial chromosome host cells is yeast cell, and it can carry up to 1000 kilobase pair. The last one are HAC, and its host cell is human cell, which can carry more than 2000 kilobase pair. Different artificial chromosomes have different methods to enter the cell. I will be talking about the yeast artificial chromosome. The first, the YAC vector is cut by two enzymes to become two fragments. Then the desired human DNA is cut by the restriction enzyme to become large chromosomal fragment. Then these two is combined together by recombination and DNA ligase. Next, the YAC will have the insert human DNA. The YAC is then put into the cell by transformation for further cloning. There is an advantage of using artificial chromosome as vector instead of natural chromosome. First, the level of expression can be increased depending on the site of insertion. The artificial chromosome expression system had developed based on a pre-engineered AC with multiple recombinant acceptor sites. This system allows for the target transfection of single or multiple genes and eliminate the need for random integration into host chromosome. And the ACE system generates stable and high expressing cell line. Next, the artificial chromosome no require the insertion of sequence into natural occurring chromosome. because random insertion of natural chromosome can potentially disrupt the important gene. The next one is artificial chromosome can clone larger fragments of DNA compared to the natural one. And the researcher also able to control the various elements of the genetic sequence. But there is disadvantage also in using artificial chromosome. Artificial chromosome will be difficult to enter the cell as the chromosome sometimes is too large. And the AC also very fragile and unstable because it is very long sequence of chromosome.
That's all for our updating slide. Any question you can ask us in the Facebook. Thank you for all your listening. Bye bye.